Assalamu alaikum everyone. So today in this video we are going to do the renal physiology. So actually I'm going to teach you the pharmacology, but before doing the pharmacology we're going to do the physiology in very quick review. So first of all we're going to do different functions of the kidney. So functions of the kidney are: it is going to be responsible for the acid base and electrolyte balance. It is going to be responsible for excretion of different metabolites from the body, and it is going to be responsible for activation of vitamin D, renin, and erythropoietin. Then 180 liters per day. Uh, 180 liters per day GFR is going to be formed, but in that only 1 to 1.5 liter is going to be excreted from the body. Okay, now we're gonna have the physiology of different parts. So first of all, I'm going to tell you the parts, and after that we're going to do different parts physiology. So first of all, we're going to have different parts in the kidney, which are the proximal convoluted tubule. This is the proximal convoluted tubule. Then we're going to have the loop of Henle. Then we're going to have the distal convoluted tubule, and in the end we're going to have the collecting duct. Now we're going to do the physiology of each part. So first of all, we're going to do the physiology of the proximal convoluted tubule. So just remember, this is the cell, this is the cell, this is the luminal membrane, this is the luminal membrane, this is the cell, and this is the interstitial membrane or the basal membrane. Okay, now this is interstitium and this is lumen. Just remember this diagram, and you can also make this diagram yourself, and it's going to be really easy for you to understand it. Okay, now we're gonna do the proximal convoluted tubule. So in proximal convoluted tubule, we're gonna have two parts, the early part and the late part. So in early part, we're gonna have different kind of channels, which are the sodium amino acid co-transporter and sodium glucose co-transporter. It's gonna go inside the cell. Okay, then in the early part, we're going to have on the basolateral membrane, we're going to have the glute channels as well as the sodium potassium ATPase, which will the sodium potassium ATPase will do, like, sodium will go towards the interstitium and potassium will come in the cell, and from the cell is going to go outside in the lumen, and then it's going to be excreted from the body. Okay, that's done. Then in late part, we're gonna have different kind of channels, which are first we're gonna have sodium chloride co-transporter, which is going to cause the absorption of the sodium as well as the chloride into the cell. Then we're gonna have the sodium hydrogen counter transporter, which is going to absorb the sodium, reabsorb actually into the cell, and secrete the hydrogen. So this is the counter transporter, as well as the water is also absorbed in this portion. Then we're gonna have the descending loop of Henle. So the descending loop of Henle is the part which is going to be only permeable for water, and it is because of absorption of the water in high concentration in the descending loop of Henle is going to make the lumen hypotonic. Okay, now we're gonna do the ascending loop of Henle. In ascending loop of Henle, this is the question which come in the paper, and it is really important, and you all should know really well. Okay, in the ascending loop of Henle, we're gonna have the channel called 1 sodium, 1 potassium, and 2 chloride co-transporter, which is going to reabsorb these molecules or these substances into the cell, and then from the cell it's going to be absorbed to the basolateral membrane into the interstitium. Okay, now apart from that, we're going to have the sodium hydrogen counter transporter, which is going to absorb the sodium inside and secrete the hydrogen outside. Okay, and on the basolateral membrane, we're going to have the sodium potassium ATPase, which is going to absorb the sodium as secretes the potassium. Okay, the ascending loop of Henle, it is going to absorb the calcium and magnesium, like the bivalent substances, through the paracellular pathway. Like between two cells, there is going to be distance, and these substances are going to absorb from there. Okay, now we're going to have the early distal tubule. early distal convoluted tubule, you can also call that. Here we're going to have the sodium chloride co-transporter, which is going to absorb the sodium and chloride, and there's going to be sodium potassium ATPase on the basolateral membrane. Okay, now we're going to do the late distal tubule and the collecting duct. Okay, so in late distal tubule we're going to have two type of cells, the principal cell, and this one is the intercalated cell. Okay, so principal cell and intercalated cell. In intercalated cell we're gonna have two types, intercalated cell A and intercalated cell B. So the intercalated cell A is going to be used in acidosis, while intercalated cell B are going to be used in alkalosis. Okay, now what happened in the principal cells, here we're gonna have the sodium is going to absorb the potassium and it's going to be secreted, and on the basolateral membrane we're gonna have the sodium potassium ATPase. This is we're done with the principal cells. Now in intercalated cells, first we're gonna do the intercalated cell A and then we're gonna do the intercalated cells B. Okay, I already told you that intercalated cell A is going to be used in acidosis. Why? So the reason behind this is, for example, this is the diagram of intercalated cell A. What happened in the cell, the water and the carbon dioxide is going to be combined together and they're going to form the bicarb. They're going to form carbonic acid. The carbonic acid is going to be break and form the hydrogen and bicarbonate. Then bicarbonate, now in acidosis obviously we have to excrete the hydrogen ion from the body. So what's going to happen, the hydrogen is going to be excreted from two types of channels. First channel is hydrogen ATPase, which is going to secrete the hydrogen, and second we're going to have the hydrogen potassium counter transporter, which is going to secrete hydrogen and reabsorb the potassium. Okay, so this is the first transporter which is going to reabsorb the potassium, while the rest of the transporter were only secreting the hydrogen, secreting the potassium from the body. Okay, but this is the first transporter which is reabsorbing the potassium. Okay, then what happens with the bicarbonate? The bicarbonate is going to be absorbed into the interstitium by a transporter known as the bicarbonate chloride co-transporter, which is going to absorb the bicarbonate and secrete the chloride ion. So this is the thing for the intercalated cell A. And if you want to know about the intercalated cell B, so just reverse this diagram. So this part will come over here and this part will come over here. So the bicarbonate is going to be secreted while the hydrogen is going to be conserved. That's it. Okay, now we're left with only one part. This is the collecting duct. So the collecting duct is a place where aldosterone and the ADH will act, and aldosterone obviously we know is going to absorb the sodium ion and the conservation of the sodium, while the ADH is going to absorb the water. The ATH is going to act on medullary collecting duct. That's it. In the next video we are going to do the pharmacology of each and every part. Thank you.